Hello, dear colleagues. My name is Yuriy Shevchenko, and I'm the Vice President of Clinical Test Innovation at Semaphore, a digital health company. We collaborated with Twist Biosciences on two of our products, and today I wanted to talk about the implementation of Twist technology in the clinical genomics space. First, let me introduce you to Semaphore. Our company has spun off from Mount Sinai School of Medicine in New York. Semaphore incorporates a clinical laboratory that used to be a part of Mount Sinai. We draw a lot of resources and enjoy productive communication with the Department of Genetics and Genomics at Mount Sinai School of Medicine. And we characterize ourselves as a patient-centered health intelligence company, advancing healthcare through data-driven results and insights. We combine high-throughput genomic testing in our laboratory with advanced analytics and machine learning tools to draw the added value from the results of clinical tests. Our primary interests are in women's health, oncology, biopharma collaborations, and lately in COVID testing and research. We combine a high-volume clinical lab and the latest in digital genomic data research. Specifically, in the field of oncology, we offer products united under the brand of Semaphore Signal. This includes the germline product, essentially a hereditary cancer test, which can be paired with somatic testing. The comprehensive molecular profiling test, looking at whole exome and whole transcriptome, as well as other aspects as needed. And of course, the bioinformatics tools to analyze the results and create a relevant clinical report. We're working with Twist on two of the products, both on the germline and front and on the whole exome, as a part of our cancer profiling test. Here's a couple of slides on the hereditary cancer panel. So from the total of 112 genes, we carved 17 genetic panels pertaining to specific conditions so that physicians could order the most relevant test as they see fit. Our interpretation comes through both knowledgeable analysts, our employees, and through our sophisticated analysis tools. This is combined with so-called Traversa platform, which allows expansion of the data knowledge and provides biobanking for the future testing needs. The next slide illustrates the breadth of our hereditary test offering. As you can see here, there are plenty of panels which can be customized even beyond this level if need be. As you can imagine, behind this variety of genes and panels, there's some serious effort on the part of our clinical test innovation team. People in the audience know very well that any probe capture product goes through multiple iterations before it becomes, perfect and covers all the requested targets. With Twist, we went through two additional iterations. First design was almost good. Then we designed a spike in to make it nearly perfect. And finally, a few more probes completely satisfied our needs and covered all the targets that we were out there to cover. Similarly, we had to go through various conditions for hybridization, washes, explore various hybridization temperatures, buffer compositions, and the duration of the hype. You can see the summary of our efforts in this slide. We used both the standard hybridization conditions, or whatever Twist called standard at the time. So the non-viscous so-called original buffer or standard buffer and the overnight hybridization. And then we used a variety of temperatures and conditions with the so-called fast hybridization buffer, as you can see here. We looked at multiple parameters, mostly through Picard HS metrics, and some of these parameters are in the table. What we're trying to achieve is a perfect balance of bases mapping to the target or selected bases, right? Minimize the percentage of duplicates, which is mostly the function of the library prep. But if you see here and analyze it a little further, it's part of capture conditions as well. We use Kappa HyperPrep, Hyper Plus, in fact, for the library construction. We've looked at various options and stopped with this one as sort of performing the most predictably and reliably in our hands. We also try to look at this parameter, which Twist is very proud of, the fold 80, which basically speaks to how even the resulting distribution of the reads is. I have to say that these are some exceptional fold 80 values. And the only caveat to that is we're really dealing with a panel of 112 genes, so we can design and balance and even cherry pick probes. As you can see, quite surprisingly for us, the best condition that we chose turned out to be a combination of the fast buffer and slow hybridization of 16 hours. This worked repeatedly best in our hands, so we stopped with that. Now, I want to mention an important point. We're a clinical genomics lab accredited in the state of New York. So I'm sure you've heard about CLIA requirements and CAP accreditation and so on. And so you likely know that New York State Department of Health requirements are the highest degree of lab scrutiny. Many labs choose simply not to certify their tests in New York State, because that will require a different level of record keeping, sample traceability, and so on. I can tell you should see the extensive binders describing each of the tests on the shelves in our lab. Of course, if the clinical test is not approved by New York State, you can't offer it in New York unless you're the sole provider of such tests. Well, all of our tests are New York certified. On the positive note, that means that we provide very high quality tests. On the negative, it takes time to jump through the hoops. So sometimes New York State Department of Health would take more than a year to issue a permission. And this is why some of the products mentioned in this presentation are over a year old. And sometimes it takes that much time to develop a test and get it New York certified. In light of that, my apologies to Twist. I'm not talking about their latest and greatest products, but mostly about the clinical products available, or our clinical test is based on the products that were available at the time of the development. And I will mention a little bit the new versions of these products. Okay, now let me talk a little bit about the somatic aspect of Signal cancer offering. Several options are available depending on the client and their interest. We offer a wide scope whole exome, whole transcriptome profiling, as well as a narrower scale pan-cancer test, which is more clinically directed at the most relevant genes. We can add MSI profiling to any or all of the above. And we definitely spent a lot of effort selecting the best exome for our whole exome test. So now on the whole exome test, we're dealing with FFPE samples, and we need a high degree of sensitivity. So if in the germline mutation scenario, we're only looking at 50-50 allele distribution, here we need to go as low as 5% or even below that. Consequently, in order to detect 5% minor allele fraction, we have to sequence very deep, so we targeted 250x average coverage of the exome. The more balanced the capture is, the less sequencing data you need to throw at it to achieve high coverage across the target regions. So we really want more of the data to be utilized. And this slide is dedicated to the graphical representation of usable depth as a function of sequencing depth. So if you can see, as you can see here, Twist exome utilizes total data better than the competitor. And again, two caveats. This is the original twist exome, not the enhanced twist exome that's available now, and it's being compared to a product by the competitor that was very common at that time. Both companies have new versions of exome products, which you're welcome to compare in a similar way. We are looking at the latest version of twist exome for the next generation of our product. So another way to look at the depth distribution across the exome is this density plot. It looks at the depth distribution in somewhat normalized fashion and uses the smoothing function for sort of better presentation. But the main point here is probably better illustrated in the right column here, where you can notice that the depth distribution of twist is closer to normalized depth. The peak is quite symmetrical around that maximum, while the competitor has a much wider peak with more of a shoulder or heavy tail. When running HS metrics, this roughly corresponds to looking at the average and median depth. So as you know, the close, the averages to the median, the tighter is the distribution, the tighter is the depth. And yet another parameter by which you may choose to look at it is the famous fold 80. This all goes to say that in this sort of bake-off, we chose twist because it provided depth that's rather uniform and therefore looks better than the competitor. An important point, and really the highlight of my talk, is that both products, both the germline and hereditary cancer panel and the somatic whole exome, whole transcriptome product, were robust and rigorous enough that they received an approval from the New York State Department of Health. Following all these stories about regulatory process that I told you earlier, this is the highest level of approval for a lab-developed test. Naturally, there are a lot of academic places where twist products are used for academic research. Now, coming from the clinical background, I would like to use that New York State Department of Health approval to vouch for twist and twist products as being high enough clinical grade to be used in a clinical laboratory. As you can see, there was some difference in how long each approval took. This is basically proportional to the product complexity as well. And of course, we now proudly offer both tests in the clinical setting of our genetics lab. Since Semaphore is a digital health company, I absolutely must plug our Bioinformatics, specifically the aspect that accompanies the signal cancer testing. We rely on our call system that we call Centralis for things that go far beyond the result to sort of the straightforward results to give the interpretation of a given test. This includes large centralized knowledge base covering thousands of variants, state-of-the-art genomic profiling of individual patient results, scientific interpretation of variants, matching patient to the available clinical trials, and finally, the integration with hospital systems. With that, I come to the conclusions slide. Our company, Semaphore, is collaborating with Twist and uses their panel design and probe synthesis for the two cancer genetic products in our New York State certified CLIA lab. We've gone through a few adjustments of hybridization, temperature, and buffer composition and arrived to a targeted hereditary cancer panel that performs well in our hands. This is based on Twist design and synthesis. Whole exome from Twist compared favorably to competitor's product and was chosen to be the platform for our whole exome somatic test. Both hereditary and somatic cancer tests received certification from New York State Department of Health and are currently offered through Semaphore Clinical Lab. This work, of course, would not be possible without uh, my team of test innovation scientists listed here and Semaphore colleagues from whose advice I benefit on everyday basis. Thank you very much to all of my Semaphore colleagues. Thank you very much to you, the listeners. I close with Semaphore slide and my email and look forward to your questions.